So this is one of the dot points. The limiting factors that could change the photosynthesis, the rate of photosynthesis. There are 4 factors. Think about the light dependent and light independent reactions and write them down. There are 2 factors in light dependent reaction. So there's a main input in light independent. And then there's another factor affects the enzymes, right? So I'm going to take the light intensity as the first one. Write down the stage that this is going to affect, light dependent or light independent. And then I'm going to take the carbon dioxide concentration, temperature, write down the stage. Is this going to affect the light dependent or light independent? Temperature. Always take the 4th one, the last one is water, right? So this is the main input of water, light dependent, but don't take this as the main factor, right? So this was not even in the study design last year. They put it in some reason, but just keep it as always the last point. Right, so light intensity, light dependent, carbon dioxide, light independent. Temperature means something to do with enzymes, right? So throughout the photosynthesis, there are enzymes, but we went through specifically the enzymes, the Rubisco and PEP carboxylase. So if you want, you can take it as light independent, the Calvin cycle. Water is mainly the light dependent because carbon dioxide, water, they are the main inputs, right? So light dependent water is the main input, carbon dioxide, light independent reaction. Let's take the light intensity. Can you write down the reaction? The water split using the energy, sunlight. And then you have to know what happened. The specific graph. Label the axes first. We are changing light intensity. So light intensity needs to be the independent variable, x-axis. If I change the light intensity, what happened to the rate of photosynthesis? Write down the reaction now. We're going to split water molecule with the use of sunlight or light. And if I split the water molecule, the main output needs to be the energy in the form of ATP and NADPH. Right? You can ignore oxygen if you want because it's just a byproduct. Right? We don't need oxygen now. So if I have lots of light, I can split a lot of water molecules, giving or producing more energy, which can be given to the light independent reaction. right? So light independent is the carbon dioxide to glucose. So if I have lots of sunlight, I can split a lot of water molecules, producing energy required for the light independent reaction to produce more glucose. So can you write down what happens with more sunlight? So it's going to split more water molecules, produce a lot of energy, right? So energy production is going to increase, which is going to increase the glucose production because there's lots and lots of energy going into the light independent reaction, right? So the rate of the photosynthesis is going to increase. So you have to know how to explain A to B, why the rate of photosynthesis is going to increase from A to B, right? So how? Higher the light intensity, higher the number of water molecules we're going to split, producing higher number of ATP and NADPH required to produce more glucose. But rate of photosynthesis won't go up. When it comes to like a maximum point, it's going to go like this, right? So this is same as the previous enzyme graphs, right? So this is because there are limiting factors. Right? So can you explain why B to C is going to be constant? What could be a limiting factor? You can write down multiple limiting factors. So B to C is going to be, the limiting factors are any other variable other than light intensity, right? So it could be the carbon dioxide. It could be water molecules not having enough carbon dioxide. Carbon dioxide is the main thing, right? So other variables going to be limiting when you exceed it beyond B, point B. Right, so from B to C, the graph is constant because of other limiting factors like carbon dioxide. Can you identify a limiting factor for this region here? So if I say something in between A and B, like this point here, write down what is the limiting factor for this point, the X. These are the questions. Huh? They might just ask why the rate of the reaction is constant from B to C because of other limiting factors, right? Other than the light intensity, your x-axis. But if there is something before, always the limiting factor is what you're changing, right? Because the light is not to the optimum. So the light intensity is the minimum limiting factor from A to B, right? When it comes to B, the light is, the maximum amount of light is receiving, but other factors are limiting from B to C, but before that, always the x-axis. Right, so you can read the x-axis. So it's not the optimum, right? So if I say light intensity is 10, the maximum amount of light the plant needs is 20. It's not 20 yet, right? So anything before A to B is the x-axis, the limiting factors, but beyond the saturation point, it's always other limiting factors. Is this clear? These are the questions, huh? Even for enzymes, they ask before the saturation point or after the saturation point. You have to know the limiting factors. Right? You can write down water if you want, but I don't think water is always going to be a limiting factor. Plants have plenty of water available, always. Is the light intensity clear? So this is it for the light intensity. Let's take the next one, temperature. Okay, I'm going to go to the next one. Let's do the temperature. No, let's do the carbon dioxide. Can you plot the graph? It's the same shape as the light intensity. Right? So always what you're changing is the x-axis independent variable. So carbon dioxide concentration. If I change the carbon dioxide concentration, what happened to the rate of photosynthesis? Plot the graph. You have to go from A, B, and C. There has to be a saturation point again. So the state that's going to be affected is the light independent, right? So more carbon dioxide coming to the Calvin cycle. You're getting lots of carbons to incorporate into glucose. Right, so carbon source for glucose is the carbon dioxide. Higher the number of carbon, higher the number of glucose molecules. So if I keep increasing the carbon dioxide concentration, up to a certain point, the rate of the reaction is going to go up. And then there's the saturation point. The rate of the reaction is going to be constant, exactly like the light intensity. Right, so I'm using the same letters A to B and B to C. So you need to know how to explain A to B, why the rate of the reaction photosynthesis is increasing, and why B to C, the rate of photosynthesis is constant. Right, always there's going to be other limiting factors. Right, on the limiting factor that's going to come after B to C. Right, it's exactly the same thing. Can you identify a limiting factor for A to B? Anything before B, right? So if I say somewhere here, limiting factor is we don't have the maximum amount of carbon dioxide. This is the maximum, right? So if you go below the maximum carbon dioxide is the limiting factor. But beyond the maximum carbon dioxide, there are the other limiting factors, right? It could be light intensity. Is this clear? So if I go back, for the light intensity, you can't increase it like too much light. It's going to kill the chlorophyll pigments. Huh? So you can't just go down after a certain point. It's just going to just destroy all the chlorophyll pigments, right? It's going to burn the leaf. Remember that, right? So it can just come down like the temperature. So this is the carbon dioxide. You have to know the limiting factors. Are there any questions? Okay, next one. Let's do the temperature. This is the temperature. So we use two enzymes in like independent reaction, the Calvin cycle. Write down the two enzymes. There are the enzymes, but these are the two you learned, right? So the main enzyme in every single plant is the Rubisco. Write down the plant types. Is it going to be in C3, C4, or CAM? And then we went through another enzyme, PEP carboxylase. Write down the plants we're going to find these enzymes in. Rubisco is the main enzyme for the Calvin cycle, right? So without Calvin cycle, no glucose. So Rubisco needs to be present in every single plant. But PEP carboxylase is just adapted to the high temperatures, low humidity. So it's only going to be present in C4 and CAM, right? So when you're considering an enzyme, it needs to be always, the main focus is the Rubisco. Okay, plot the graph. The enzyme, the temperature graph, right? The normal one. So go with the optimum temperature. So can you use all the keywords we went through when I did enzymes and explain why the rate of the reaction is low in this blue region, and then explain the beyond optimum? You have to use the keywords, huh? So anything less than optimum temperature, the keywords you have to use the Rubisco, use active site, use kinetic energy, and use number of collisions, right? And then compare the glucose formation. Use all these keywords and write down the reason why the rate of reaction, you can take whatever the temperature, like around 10 degrees, why the rate of reaction is low if the temperature is less than the optimum. Use all the keywords. Okay, you have mentioned substrates, huh? Remember the Rubisco substrate is RUBP, right? Or you can use the term substrates, right? RUBP and carbon dioxide, right? So when you mention in the kinetic energy, the Rubisco and the RUBP carbon dioxide, they're not, don't take the gas. RUBP, they're not having enough kinetic energy. So less collisions between the Rubisco's active site and the given substrates. That means less amount of glucose produced. Also finish the 10 degrees. Write down why the rate of reaction is less if you go beyond optimum. Give me a minute, I'm going to write down the keywords. Huh? So you have to mention the Rubisco active site, whether it's present or not, and denature. So if I increase the temperature beyond the optimum temperature, the Rubisco is going to denature. So the denatured Rubisco won't have active site, so the substrate can't bind. Less or no glucose production. Right, so if you ask for the optimum condition, you go with the kinetic energy, the collision is the maximum, right? So the stage mainly affected is the Calvin cycle or light independent. Light independent. Let's do the last one, water availability. Everyone finish? Okay, let's go do the next one. Water availability. Write down the reaction we need the water for. So the light independent reaction, the main source is water. We're going to split it using the sunlight to get the energy for the light independent. Right, so you can write down if you want higher the water, higher the amount of energy we're going to produce. But the amount of water needed for the photosynthesis is less than the amount of water needed to sustain the life. So there's going to be lots and lots of water already stored inside the plant cells, because plant cells also always needs to be turgid. Can you remember this term? So if you have like a normal plant cell, if it is like a well-watered plant needs to be under turgid conditions always. If the plants are under turgid conditions, means there's plenty of water already available inside the cell. Right? So a lot more water needed by the plant to sustain the life than the amount needed for the photosynthesis. Right, so if you compare the amount of water needed to sustain the plant life is much more higher than the amount needed for the photosynthesis. Right, so most of the time water won't be a limiting factor, right? Main limiting factor. Before that, the plant will die. Is that clear? So that's why I said don't write it as the first point, right? If you ask for only 3 points, go for the main ones like carbon dioxide, light intensity, carbon dioxide. Because plants will always maintain enough water to sustain the plant life, right? So compared to that amount, the water needed for the photosynthesis is very less. But you can think about the indirect effect of water. So if in case the transpiration is, water evaporation is very high, right? If the water is not available, right? That means if the water is just evaporating through the stomata, right? So the water is evaporating, lots and lots of water evaporating through the stomata. Because of this, the stomata will close. Then we have the problem because inside the plant leaf, they're going to use all the carbon dioxide and the plant won't have enough carbon dioxide for the Calvin cycle. Right, so if we consider a C3 plant will shift to the photorespiration. Right, so carbon dioxide, no carbon dioxide means less glucose production. So there is an indirect main effect to with the water availability. Is this clear? Right, so if there's no carbon dioxide, no glucose. I'm going to start the cellular respiration. That's a CRISPR-Cas9 part again. CRISPR, how it is going to increase the crop yield and productivity. I'll do it next week. Okay, we'll start the cellular respiration. But I'm going to, so I did the photosynthesis. And I'm going to start the cellular respiration now. Right? So I will finish it by next week. That means I'm going to give the test on 10th of March. Can you write it down? I'm giving 2 weeks to study. Every single thing, right? C3, C4 CAM. Whatever I'm doing for cellular respiration, it's the same as the unit 1 stuff. But we did a lot on photosynthesis. Just study every single thing. (10th of March, huh? Okay, write down the new topic, cellular respiration. This is easy. This is exactly the same as last year. Okay, can you draw the mitochondrion first? Can you remember the matrix? Cristae, inner membrane, and outer membrane. Just label everything inside like a big diagram. Something like this size. So outer membrane is flat. But the inner membrane, there's lots of inner membrane means the site of the electron transport chain, right? To give the enough space for the electron transport chain, it needs to have more space. So it's not like a flat membrane like the outer membrane. You're going to see these foldings, right? Because of this, it's going to increase the surface area for the electron transport chain. So inner membrane is always longer than the outer membrane. And the liquid is the cytosol-like liquid is the matrix. And the foldings, these highlighted parts are the cristae. right? So the cristae is made out of inner membrane. So I'm going to just highlight one, and this is the cristae. Right, so you have to know the details of the photosynthesis, the chloroplast, and the mitochondrion, right? And then you have to know everything on the protein secretory pathway, the RER and stuff. Write down the function of the mitochondrion. Use the term cyto. So you can say site of cellular respiration. Or you can say site of ATP production. Okay, so this is the mitochondrion. Let's start the cellular respiration. Write down the balance equation. It always balance the equations, don't write unbalanced ones. Don't have to write ATP, write down the breakdown, just opposite of photosynthesis. So from this mitochondrion, you need to know matrix and the cristae, yeah? Because cellular respiration reactions in matrix and cristae. Right? Krebs cycle in matrix, cristae is the electron transport chain. Yeah, remember, it's exactly the same as last year. Write down the balance equation. So cellular respiration is, we are taking the 6-carbon glucose and using 3 stages, we're going to break it down step by step until it produces carbon dioxide. Right, so this is a catabolic reaction, this is an hexagonic reaction. If you go from big molecule, which is 6 carbon, step by step, if you break it down until we get 1 carbon dioxide, energy going to be released. Right, so cellular respiration main purpose is to collect ATP in every single reaction. Because this is hexagonic, the energy release needs to be trapped in the form of ATP. For this, we are using oxygen, which will become water. Right, if you want, you can change the colors. Oxygen is the one that's going to produce the water molecule. Right, so glucose becomes carbon dioxide, oxygen will become water. Can you balance it? Same as the photosynthesis. So every single organism need to have glucose, including animals and plants, so they can undergo photosynthesis. They can undergo cellular respiration to produce their own ATP. Right, so always cellular respiration, the substrate is glucose. Write down how the animal is going to get their, let's start with plants. Write down how the plant is going to get their substrate for cellular respiration. Right, so how the plant is going to get their glucose, because any living organism, if they are living, they must produce the energy, right? So the higher organisms, plants, animals, they have the cellular respiration. How the plants going to get their glucose? How animals or humans going to get their glucose? We did the digestive system last year. Okay, write down the photosynthesis. Cellular respiration. The glucose is the source. So plants undergo photosynthesis just to produce glucose for the cellular respiration, right? So the source for this glucose is the photosynthesis. The product of the photosynthesis is the input for the cellular respiration. Right, so this is it for the autotrophs. Plants are autotrophs. Remember, I used the term autotrophs. They can produce their own organic compounds like glucose. But if you consider any heterotroph, including animals or animals or humans, they have to use the existing organic compounds produced by the plants. So as an example, if I eat a big starch molecule, I'm consuming lots and lots of glucose. So this is how the starch is going to look like. It's just the combination of lots and lots of glucoses. But inside the digestive system, your digestive enzymes will break down the bonds between glucose and release individual glucose. Right? So we get the substrate, but we're going to just depending on whatever the other organisms that already made the organic compounds. Right? So whether we can produce by ourselves or we can get it from the other organisms, the substrate needs to be available for the cellular respiration is the glucose. Right, so plants can make their own glucose, but we can get the existing other sources of carbohydrates and break it down to get our glucose. Right, so this is the glucose that can go through the cellular respiration. Let's do the cellular respiration. Write down the 3 stages we did last year. Exactly the same, and write down the location. Right, I drew a cell, so you can see the mitochondria and the cytosol, right? So I have glucose as the main substrate. 6-carbon glucose. I'm using 3 stages to break it down until it becomes 1 carbon dioxide. Right, so the first stage is the glycolysis. Write down the location. Second one is the Krebs cycle. Write down the location. Last one is the electron transport chain. Or the system, write down the location. So you have to add the mitochondria, right? If it is part of the mitochondria, you have to have the mitochondria written. So the glycolysis is in the cytosol, not inside the mitochondrion yet, right? So the first breakdown is out of the mitochondrion. But the Krebs cycle and electron transport chain is going to be inside the mitochondrion. Krebs cycle is the liquid, the matrix. Electron transport chain is in the mitochondrion cristae. We got deduct marks in, I think, 2017 for not writing mitochondrion, right? I think it was matrix they were asking, right? Those who just wrote matrix didn't get any mark. So don't lose easy marks, write down always the organelle, right? So even if you say thylakoids, that's why I said it's chloroplast thylakoid. Definitely matrix is like a common term. We can't find the matrix somewhere else. So you have to say it's part of the mitochondria, right? Even the cristae, say somewhere it's part of the mitochondria. Let's do the glycolysis first. So we are not inside the mitochondrion yet, we are in the cytosol. So I'm going to get the glucose. We started with 6 carbons. And we're going to split it into half. Write down the number of carbons. 3 carbon each, but you produce 2 of them. Write down the name, it's the same thing last year. You produce 2 pyruvates. But this is an exergonic reaction, release energy. Right, so energy is going to be in the form of ATP. Cellular respiration collects ATP, so you must know the number, right? It's a must. So the first stage is producing 2 ATP. So when the energy is released, energy is going to be also trapped in the high energy protons or electrons. And this is going to be taken by, we can only use 2 coenzymes, right? So for photosynthesis, we use NADPH. The loaded one is NADPH, but we are doing a cellular respiration. So we can use the NADH or FADH2. Right, but for the glycolysis, we are using only the NADH. Don't write the FADH2 yet, right? So can you go and grab it with the unloaded coenzyme and make it into a loaded coenzyme? If you want, you can add the plus. Right, so unloaded coenzyme goes and grabs the high energy proton and becomes the loaded version. Right, so the main purpose of doing the cellular respiration is to get the energy, right? So I have two ATPs, the usable source is the ATP, I have two at the moment. Write down inputs and outputs. 3 inputs, 3 outputs. Start with outputs. Start with the carbons first and then go to the energy. Finish. Always start from the carbon. So the pyruvate coming from glucose. And we have ATP and NADH. ATP come from ADP and PI. If you want, you can add 2, right? No need. NADH came from unloaded coenzymes. Right? If you know this 2 ATP, more than enough. Don't add 2s or any numbers other than that. So I have partially broken down the glucose. I started from 6 carbon glucose. And I'm going to break down glucose until I see 1 carbon dioxide. But I have only partially broken down the glucose because it's not carbon dioxide yet. It's something called pyruvate. That means we can further break down pyruvate, but every single stage you're going from big molecule to the smaller, releasing ATP, right? We have 2 ATP at the moment. We're going to get more ATP when we break it, the pyruvate, into 1 carbon dioxide. Right, so first stage is done. We're going to go to the next one. So this part is done. Now I'm going to take the pyruvate and break it down further. Right, but each and every stage we are going from big molecule to small, releasing energy form of ATPs. Right, so let's do the next one, Krebs cycle. Write down the location. So if I draw the cell, pyruvate is the molecule. So we broke down the glucose into pyruvate inside the cytosol. But pyruvate is the molecule that now enters into the mitochondria. So when we draw the Krebs cycle, it needs to be a cycle like Calvin cycle. So can you just leave some space and draw a circle? Exactly the same as the clockwise cycle. But the input of Krebs cycle is a 2-carbon acetyl coenzyme A. Right? So write it down. What enter into the Krebs cycle is the acetyl coenzyme A. It's a 2-carbon compound. So if this is the Krebs cycle, if the acetyl CoA enters into the Krebs cycle and breaks down until carbon dioxide release, anything happens before has to be pre-Krebs cycle. Right? Because Krebs cycle starts from the acetyl coenzyme A. Whatever happens before needs to be pre-Krebs cycle. Right? So pre-Krebs cycle means you're breaking down the pyruvate into acetyl CoA. If you want, you can use the acetyl CoA. Otherwise, you can just ignore acetyl CoA and say pyruvate enters into the Krebs cycle. Doesn't matter. Right? But I know some schools use the acetyl coenzyme A, so you can use it. Otherwise, just say, forget about the pre-Krebs cycle. Just think like pyruvate is the one that enters into the Krebs cycle. Either one is correct. Right? But this is what actually happens. So I'm going to leave it like this. So pyruvate enters into the mitochondrion and break down into acetyl coenzyme A. So this is a 2-carbon compound. And then acetyl CoA enters the Krebs cycle, further breakdown until it releases carbon dioxide. Right? So we went from the glucose 6-carbon until the last carbon dioxide. Can you draw an arrow going out and write down carbon dioxide? But still, this is an exergonic reaction. It's going to produce energy, right? So energy is going to be in the form of all 3 forms. I'm going to draw the arrows. Can you write down the type of energy we are getting here? ATP, you have to know the ATP number, only 2 ATP. Output needs to be the energy, right? So arrowhead needs to have the energy. So we have 2 ATP. And all the high energy protons going to be grabbed by NADH. and FADH2. Right? Write down the unloaded ones. Right? If you want 2H. Can you write down inputs and outputs? You can say input is the pyruvate, right? So forget about the acetyl-CoA. Write down input is the pyruvate. Start from the output or the inputs. You can see all four. So if I have the pyruvate or acetyl-CoA as the input, I'm writing a pyruvate. The output is, because this is the carbon source, output has to be carbon dioxide. Right? So the other types of outputs, they are the energies. So we have 2 ATP. Don't write the number here, no need. ATP, the coenzymes NADH, and the coenzyme FADH2. Then go for the unloaded. This is clear? These are study design dot points. Now you have to know inputs, you have to know outputs. Right? Finish? Latika, finish? Okay, let's do the last one. Last one is the electron transport chain. But I started breaking down glucose. because I can step by step break it down until I produce carbon dioxide, the one carbon dioxide. So when we go to the end of Krebs cycle, we have fully broken down the glucose because Krebs cycle produced carbon dioxide already. But during the glycolysis and Krebs cycle, we have produced the different coenzymes. We have the NADH and FADH2s. Right? So remember the example, the ATP is the only usable energy. Any other source of energy, before we use the energy grab inside these enzymes, it needs to be first converted into the usable ATP. Right? So the next one is the electron transport chain. We are not going to break down any carbon compound, right? Because carbon being fully broken down from the glucose until carbon dioxide. But we have still collected lots of coenzymes like NADH and FADH2 throughout glycolysis and throughout the Krebs cycle. So we won't just keep it as these other coenzymes. We have to convert it into the usable energy type ATP. So we have to do the electron transport chain to convert all the other coenzymes back to the ATP, the usable energy type. Right? So there's no carbon breakdown in electron transport chain. It is just the conversion of other coenzymes back to the usable ATP. Right? So let's do the electron transport chain over the system. So you can say ETC or ETS system, right on the location. Mitochondrial cristae. Right, so I'm going to just draw part of cristae. I'm going to enlarge this part and draw like a massive cristae. So let's draw this the cristae. So this is the outer membrane and this is the inner membrane. I'll come back to this slide now. So normally mitochondria are going to have lots of proteins inside, right? Draw the mitochondria again. Along the cristae, there's going to be lots and lots of cytochromes, right? We're going to use cytochromes again and again and again. So just draw some cytochromes along the cristae. Right, so these are the proteins, the cytochrome proteins. There are different types of cytochromes, but if you can at least write down cytochromes, would be enough. There are cytochrome A, cytochrome B, cytochrome C complexes. This overall, right, you're using cytochromes for this electron transport chain. Write it down, cytochromes are proteins. Protein means you can affect the proteins like the normal, whatever the things, right? Proteins can become denatured, it can be inhibited by inhibitors, right? So highlight the cytochromes being proteins. So I'm going to draw the cytochromes. And then at the end, I'm going to draw another enzyme known as ATP synthase. So ATP synthase is the ATP making machine. ATP synthase. So when you see A suffix, you know it's an enzyme, right? So you can say ATP synthase. So that needs to be at the end of the cristae, right? So towards the end of the electron transport chain, you can, or cristae, you can add the last protein or the enzyme. Enzymes are proteins. ATP synthase makes ATP. So now I'm going to just describe about the electron transport system, right? There are exam questions asking you to explain what the electron transport chain is, right? So what happened here is, we have I said electron transport chain needs to convert these other coenzymes into ATP, right? So that's the reason of doing this. So we have produced coenzymes in the matrix and in the cytosol. So all these coenzymes come towards the cristae. Right, so they're just going to go towards the cristae and they're going to unload all the high energy protons. Right, so all the coenzymes will travel towards the cristae and unload their high energy protons. Right, so these are the unloaded high energy protons. So these high energy protons, so the first point is always you're unloading all the coenzymes, right? The NADH and FADH2. So all these unloaded coenzymes going to go through this chain of cytochromes. So along this cristae, through the cytochromes, the high energy proton is going to flow until it reaches the ATP synthase as the final protein. So can you move all your high energy protons along this cristae, but you have to go through the cytochromes, right? They're like directing the high energy proton flow. When you're writing it, you have to use the high energy proton going through the cristae, but just somewhere mention the cytochromes. Right, so the flow of high energy protons will go through the cytochromes, and at the end, high energy proton flow, so the red line means the high energy proton flow, right? At the end, the high energy will go through the ATP synthase. Can you mention the last protein enzyme? ATP synthase. Right? So the last point is cytochromes, through the cytochromes, and finally it just went through the flow of high energy protons, went through the ATP synthase. Right? All these high energy protons are going to go through. But if I say high energy proton altogether going to have energy component, and there has to be the hydrogen. Right? Somehow I need to explain the hydrogen and energy. So just think about high energy proton has both of them inside. So high energy proton has the hydrogen as well as the energy, right? So both of them together is the H plus. But now we're going to extract the energy component and separate out the hydrogen from the energy, right? So what happened is, now the flow of high energy protons finally went through the ATP synthase, and ATP synthase will extract this energy component from the high energy proton into ATP. Right? So high energy protons energy part, we're going to convert it into ATP. This is clear? I'm just using the keywords. Huh? You don't have to know the chemistry behind this because this is not chemistry. So high energy protons, I extracted, when the high energy protons go through the ATP synthase, the energy is going to convert into ATP. So we have unused hydrogens now. Once the hydrogen come out, it needs to combine with the oxygen that we breathe in. So the cellular respiration reaction has oxygen as the input, right? So this is the place where using oxygen. So oxygen is going to go and grab the hydrogen and it will convert into the final product, water. Right? So high energy protons energy component, we converted into ATP. But the leftover hydrogens needs to be accepted by oxygen and it needs to produce water. Right? I will give you all the points, but you have to mention all the formation of water. This is one of the main outputs. But the main output is ATP, right? But you have to know the oxygen grabbing hydrogen forming water. So before we list them down, the steps of electron transport chain, can you write down the inputs and outputs? I need 4 inputs, 4 outputs. So this part is a bit confusing, not like the normal inputs, outputs in other stages. Not confusing, but just think and do it. ATP and NADP, NADH, FADH2 needs to be on 2 different sides. Okay, so let's do list them down. If you forget about oxygen, you can write it down first, right? So input is the oxygen. It's grabbing the hydrogen and forming water. And the main purpose is to convert these other coenzymes into energy. So once they break down, they need to produce ATP. Right? Now you can complete the rest. So you need to know the electron transport chain, the ATP numbers. But electron transport chain is the one that makes up the biggest number of ATP. I normally say it's 32 to 36, 32 to 34. But now Vika says 26 to 28. There was a range always. If your school says 32 to 34, it's okay, right? So normally they used to say they're going to accept like a range. Yeah, we used to say 32 to 34, but different cells produce different numbers. You can say 26 to 28. Right, so I will confirm which one is correct, but there is a document says 26 to 28, right? But this is like significantly higher than the other two stages, right? So write down electron transport chain. Something happened to the electron transport chain. You're losing a massive number of ATP, right? You need to know the exact number, right? So I think this is the correct one, but we used to say this. Let's go with 26 to 28 ATP. So if something happened to electron transport system, you're losing 26 to 28 ATP, a massive number of ATP. Okay, write down the question. Can you explain the electron transport chain? In detail, explain what the electron transport chain happens. What happened in the electron transport chain for the system? 4 marks. So these are Vika exam questions. Write it down, use every single possible keyword and write down until you produce water and all the ATPs. Okay, the first one is, the first point, you have to explain the unloading of the coenzymes, the unloading ones. And then mention the 2 coenzymes you're unloading. And then there's going to be a flow of high energy protons. You have to go mention the cristae, cytochromes, so the flow of high energy protons will go through the cristae, through the cytochromes. And the last, third one is, finally it will go through the second enzyme, ATP synthase. Which will create ATP, give the ATP numbers. It's very, very, very important. Write down the ATP numbers, 26, 28 ATP. This is the maximum so far. And the remaining hydrogen will join with oxygen, the atmospheric oxygen that we breathe in. The final product is water. Right? So the final proton acceptor, write down oxygen as the final proton acceptor. So the proton is going to be accepted by the oxygen and it will form water. Right? So these are the keywords we need you to include to get the full marks. Now don't take shortcuts, you're losing it. The main thing is, I always need ATP numbers. Don't say like high number of ATP produced, you're losing the marks. You have to be really, really specific, right? Let's count the number of ATP produced. Is this finished, done? Okay, let's go through the ATP numbers. Can you write down the ATP produced in glycolysis? The Krebs cycle and electron transport chain. And then calculate the total number of ATP. So because they have changed it, this is like different to whatever you learned last year. Right? Because the electron transport chain, they reduced it. So glycolysis, there's only 2 ATPs produced. Krebs cycle, we produce 2 ATP. But electron transport system produce 26 to 28 ATP. And then write down the total. $30 to $32. So whatever your textbook says, go with 26 to 28 because there's a frequently asked questions document, the weaker one. It says 26 to 28, right? I think some textbooks says the wrong numbers here. But they used to accept a range, but now they say just because says 26 to 28. Let's go with this. Right, so this is what happens if the full cellular respiration takes place, breaking down glucose into carbon dioxide. right? So this takes place in the presence of oxygen. We use the term aerobic cellular respiration. So aerobic cellular respiration produce lots of energy. We will end up with 30 to 32 ATP. Right? Okay, I'm going to do something else. I'm going to do a question. Write down the 3 stages one more time. Don't use the abbreviations. Don't write ETC, ETS, you're losing the mark. So always these 2 cycles, the Krebs cycle and electron transport system is inside the mitochondrion. And they are like coupled reactions. And just remember, they both require oxygen, right? Or in other words, these 2 reactions are going to take place only if the oxygen is available. Right? If you need use all the oxygen in electron transport system, but Krebs cycle all needs the oxygen as well. Right? So if something needing oxygen, we're going to call them as aerobic processes. That means these 2 reactions won't take place if the oxygen is not available, because they are aerobic processes. They will take place only if oxygen is available. But glycolysis is outside in the cytosol. It does not need any oxygen. It's going to take place whether oxygen is present or not. If any reaction happens without oxygen, they are the anaerobic processes. Right, so this is something you have to remember. Glycolysis is an anaerobic process, but the other 2 cycles, the reactions happen inside the mitochondrion, need oxygen, right? So these 2 are coupled reactions, both require oxygen, indirectly or directly. So they are both aerobic processes. You have to remember this. I need you to use these terms. Right? So if I use the glycolysis, if I give you the glycolysis, you need to know it's an anaerobic process. If I give you the Krebs cycle or the electron transport chain, you have to use the terms they are aerobic processes. Okay, I'm going to share a question now. I'm going to send the first part. Read the first part, I will send you the second part. This is the question. Okay, sent. Can you answer the question? This is like the biggest question so far, 6-mark question. When you're writing the answer, this is a VCE exam question, right? Don't write the same answer twice, right? So every single box needs to have different, no, the reason needs to have different reasons. You're not going to write exactly the same thing twice. Is that clear? Okay, think and do this. Use the terms aerobic and anaerobic and think about which one needs oxygen and which one doesn't. So the first one, it's asking changing oxygen concentration, whether it's increasing, decreasing, or no change, right? Now think about, this is not photosynthesis, right? So if this is not photosynthesis, you're only going to use up oxygen, you're not going to produce any oxygen, right? So you have two options left. Either it will decrease, if it is not using oxygen, it will have no change, right? There's no, you don't have to use increase. Because this is cellular respiration never ever produce oxygen, right? So write it down for every single stage, whether the oxygen going to decrease or no change, decrease or no change. And whatever the point, you need to give the reason. Okay, let's go through this. Have a share display. Okay, can you see this? Let's start with the experiment 2, right? Because they're adding the cytosol, right? So cytosol means the place for the glycolysis, right? And then we go to the mitochondria. So start from the second one. So the first one is the glucose. They're adding glucose. So the reason is glucose is the substrate for the glycolysis, but glycolysis is an anaerobic process, right? So the oxygen concentration, there's no change to the oxygen concentration. Is that clear? So first one is no change because you have the substrate glucose, you have the cytosol, but this is an anaerobic process, not going to consume any oxygen. Is that clear? Next one. So experiment 2, the pyruvate, right? We have the cytosol, so we can only do the glycolysis, right? So for this one, you can say there's no mitochondria to break down the pyruvate, right? Or to undergo Krebs cycle and electron transport system, there's no mitochondria available. So without having the mitochondria available, the pyruvate can't break down further into any carbon dioxide. Right? So Krebs cycle and electron transport system will be the substrate for the pyruvate, but to undergo this reaction, we need the mitochondria, but mitochondria is absent. So oxygen concentration, no change. Is it clear? This is like one of the most complex questions I have seen. Is the experiment 2 clear? Okay, let's go to the experiment 1. So suspension of mitochondria is available. So we have to undergo Krebs cycle and electron transport chain, but glucose is not the substrate for the Krebs cycle, right? So the first initial reaction needs to be the Krebs cycle, but for Krebs cycle, we need the pyruvate, not the glucose, right? So we are not using glucose in the Krebs cycle, so there's no oxygen, no change to the oxygen concentration. Because we don't have the proper pyruvate, right? We don't have the correct substrate. But the next one, we have the side mitochondrion, we have the substrate. So Krebs cycle and electron transport chain can undergo. There are aerobic processes, will consume oxygen. Oxygen will decrease, the experiment 1 under pyruvate. Right, oxygen will decrease because Krebs cycle electron transport chain, there are aerobic processes. Pyruvate will be broken down further, consuming oxygen, dropping the oxygen level. Those who finish this one, can you think of now the last one? That's the most complex one. I want you to think about the glucose. So the last one, let's do the pyruvate first. I'm taking the pyruvate first. So the third experiment, they have both, right? So the mitochondrion and cytosol, that means you can do the full reaction, right? Because the mitochondrion is available, the Krebs cycle electron transport chain can undergo. So they are aerobic processes, will consume oxygen. That's for the pyruvate, right? So for this one here, the pyruvate, experiment 3, will decrease the oxygen concentration, because Krebs cycle electron transport chains, they are aerobic processes, they will use the oxygen to break down pyruvate. The glucose so I'm doing experimentally glucose, huh? The reason is because the cytosol is available, glucose will become pyruvate. But that pyruvate is going to be used in the Krebs cycle using oxygen to further break down until carbon dioxide. Is that clear? Glucose is not direct substrate for the aerobic process, right? But glucose will break down in the cytosol to produce pyruvates. But this pyruvate will undergo an anaerobic process, which is the Krebs cycle, or you can say electron transport chain. So the glucose product, pyruvate, will use oxygen to further break down until carbon dioxide, right? So the second stage going to consume oxygen. So oxygen is going to drop. This is a 6-mark question, right? So after this question, most of the schools and most of the SACs and everyone using this concept, I want you to go through this, right? Download it and go through the answers. Give me a second, I'll send you the answer. Otherwise, I don't know what you have written down. Give me a minute, I'm sending the answer. Here's the answer. Download the answer. Are you good? So read the answer, and this is like the hardest question so far. Okay, so I have little bit to do the anaerobic respiration, the endosymbiosis, and then the biofuel and that photosynthesis CRISPR-Cas9. But I'm going to finish this next week, and then you're doing the test on the 10th, right? You have enough time. I'm done. Bye.
Thank you.